Okay, so neurons connect with each other at gaps that are called synapses. And for example, here is one neuron, right? Here is the cell body with all the dendrites that are receiving various inputs, both excitatory, which means depolarizing, right? Heading towards that threshold of minus 55, right? Opening, let's say, sodium channels to let sodium come in, right? And depolarize. And then, there's the initiation of an action potential right here at the axon hillock. If you reach minus 55 here, you swing open all of those voltage-gated sodium and potassium channels, and you start a wave of depolarization, repolarization, sodium entry, potassium output, or outflow, going down, 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 propagating down this entire axon to the terminals. And this is what we're looking at in this image, is we've got what we call the before synaptic membrane, right? And there's, at that terminal swelling here, there was all sorts of specialized machinery. And then across, what's being represented is what's called the postsynaptic, membrane. This is the dendrite, let's say, of the next neuron that can be either, excited, depolarized, right? Or we're going to see it could also be inhibited, right? Hyperpolarized, made less likely to fire. So at this presynaptic terminal swelling right here, you can see a number of different, critical protein machines, many of them which could be influenced by drugs. For example, there is another kind of voltage-gated channel that you see indicated. It's, instead of being a voltage-gated sodium channel and a voltage-gated potassium channel, this one is a voltage-gated calcium channel. Calcium is another very important dietary constituent. You need to, consume calcium. And calcium also, it turns out, will swing open, will ungate at the threshold potential, or at a threshold potential of around, minus 55 millivolts again. So again, the neuron is, at the resting potential here at the end, before the arrival of the depolarizing, repolarization, wave of the action potential, the action between those two potentials. And when that depolarization hits the terminus, well, it swings open these voltage-gated calcium channels, and then calcium, which is in high concentration outside the cell, and low concentration, very low concentration actually inside the cell, because it turns out calcium is highly reactive. It'll bind to things and initiate all sorts of intracellular changes, so it's kept quite sequestered inside the cell. But when the action potential arrives, when you depolarize to threshold, minus 55, well, you swing open the gates of these voltage-gated calcium channels, and the calcium rushes in. And remember, it's driven again by the concentration gradient force, because there's more outside than there is inside, and also by the electrical gradient, right? The electrical force. It is negative on the inside at minus 55, and calcium, Ca2+, is a positively charged ion. Other important kinds of, sort of constituents that define, structural features that define this axon terminal are these round, sort of spherical, membrane-bound sacs that are called vesicles. And the vesicles are really critical because they are filled with neurotransmitter. They're going to be filled with the chemical that this neuron is going to use to communicate, with the next neuron across the synaptic gap or the synaptic cleft. And these vesicles, they're phospholipid bilayer bound, so they're very good at keeping things in versus out. So in order to get the neurotransmitter inside the vesicle, you need another protein machine, and that's indicated here too. This is what we call a vesicular transporter. So it's utilizing ATP, right? It's breaking that bond and releasing that chemical energy, sort of converting it into the kinetic, transport, the grabbing onto a neurotransmitter. We're going to see like dopamine or glutamate or something on the outside of the vesicle membrane, and it's going to pull through itself, pull that chemical, that neurotransmitter, inside the vesicle. So it's packing the vesicle tightly with lots and lots of dopamine molecules, or serotonin molecules, or glutamate, or GABA. A neurotransmitter is being packed inside the vesicles because of the energy-utilizing vesicular transporter. And what is the force that it is fighting, that it needs ATP to fight? Well, it's going to be the concentration gradient force, because you are taking the neurotransmitter, which is more widely dispersed, in the axon terminal, and you're trying to pack it and force it into high concentration inside that vesicle. So that vesicular transporter is a really critical machine here at the presynaptic membrane, of the neuron, that's going to be sending the chemical message most typically at a synapse. And we're storing up concentration gradient force, right, by utilizing ATP with that vesicular transporter. And we're going to see some drugs are actually going to influence the vesicular transporter and wreak some havoc. So you also see these vesicles that get filled with neurotransmitter, packed tightly because of the vesicular transporter. They're what they call docked with a series of proteins, and this is only indicating a few little boxes that represent proteins here at the membrane. But really, it's about 28 distinct proteins that form a docking complex that sort of pull the vesicle close to the membrane of the presynaptic terminal. And what's sort of envisioned is that the docking complex, the protein, this is a vesicle, pinches off just a tiny little piece of the vesicular membrane and brings it in close apposition, like really close to the actual axon terminal, the presynaptic membrane. And so they're really close together. They're not quite touching, but those proteins have sort of brought them close. And there's a line of them right up there against the membrane on the inside of the cell. And then there are more vesicles behind that are being, packed with neurotransmitter because of the vesicular transporter. So what happens upon the arrival of an action potential? And I want you to remember that an action potential, again, is a wave of depolarization, right? The opening of the voltage-gated sodium channels and the rushing in of sodium. The repolarization as the potassium moves out through open voltage-gated potassium channels at, the threshold potential and, down. And then the movement of the sodium into the next segment of membrane, which will depolarize again to threshold, open more of those, voltage-gated sodium channels and allow for propagation all the way down. So you're getting, basically the arrival of depolarization basically at the end point here, which will swing open the voltage-gated calcium channels, which are, present in, high density around this axon terminal membrane and the presynaptic membrane, and will allow for the sudden entrance of calcium ions into the presynaptic terminal. And what that does, the calcium that comes in, remember I mentioned it was very reactive, so it gets snapped up by something. Some chemical binds it, and that changes its shape, and it allows this kind of activated chemical to interact with the docking complex, so it fits now into some component of the 28 proteins that are gripping and holding the vesicles right next to the presynaptic membrane on the inside, and it brings the membranes closer. And what it seems to do is it causes this sort of fusion of the vesicular membrane with the presynaptic terminal membrane. So you can see there's like a little, there's still like kind of a neck held here with the vesicle behind, but now the neurotransmitter, which is packed in high concentration in those vesicles, right, has a little tiny hole out into the synaptic cleft, into that gap between the first neuron and the next. And the concentration gradient force will drive that neurotransmitter out. It'll leave because it's in high concentration on the inside of the vesicles, and in the cleft there's very little neurotransmitter, so it'll move out because of the concentration gradient force. And then we're going to see it's going to interact with proteins that are embedded in the membrane of the postsynaptic cell. We're going to see it also interacts with proteins that are embedded in the membrane of the presynaptic cell. But these proteins across, there's often, especially at excitatory synapses, like a thickening called the postsynaptic density. And that's just all these proteins that are kind of embedded in that phospholipid bilayer that are going to be receptor proteins. They're going to receive the chemical message of the released neurotransmitter, and they're going to bind it. And we're going to see that the binding of the neurotransmitter to these various types of receptors that are located, let's say, postsynaptically, well, they're going to cause a change in the conformation or shape or structure of that neurotransmitter receptor, kind of complex. And that change in structure is going to have a significant change in function, including we're going to see, depending on the type of receptor, the charge difference across the membrane.